Okay, so today I will not be showing my video because I'm not feeling well today. Anyways, today we'll be discussing about the paper that I sent you last week.
Yeah.
Also, I thought of discussing a codominance question so that you can have an idea. But afterwards, in January also, I'll have a separate day to discuss your doubts. But today I thought of doing that 'cause then that means the fear of that question will be reduced, right? Okay. So now I will be sharing the screen. Okay. Right. So this is the question that I have taken. In this question, you can see that they have mentioned human blood group is an example of a phenotype determined by alleles that show codominance. What do you mean by codominance? Codominance means, right? Codominance means if you take, now you know we have two sets of chromosomes, right? We have a set of chromosomes. For example, if you take a human, we have 46 chromosomes. But we know they are in 23 sets, or they are in pairs of 23. That means for each of our chromosome, there is another chromosome which exactly look like this chromosome. And they are called as homologous chromosomes. So let's assume in a person's body, right? Let's assume in a person's body, there are 10 chromosomes, right? It's not true. You know, there are 46 chromosomes, but I'm just assuming, right? To do this, I'm just assuming there are 10 chromosomes. 10 chromosomes means how many sets of chromosomes? There will be 5 sets of chromosomes, right? 5 sets
Yeah.
of chromosomes like this, right? 5 sets of chromosome like this, and they are going to be homologous to each other, which means they get one chromosome from their mother, they get the other chromosome from their father. Again, this from the mother, this from the father. Likewise, only the homologous chromosomes pairs are made. And you know, right, these chromosomes have genes, right? If you take a DNA, in a DNA segment, you know, a set of nucleotides like this, which codes for a protein, which codes for a protein is what you call as a gene. And you know, when the DNA is associated with histone molecules, you call it as a chromosome. So you don't have to get afraid of the chromosome. It's just a DNA molecule associated with histone molecule. It's just a DNA molecule associated with histone proteins. Right. Now, there are so many genes in a chromosome, right? There are so many genes, right? Each of these genes code for different proteins. For example, I'll say this gene codes for hair color, and this gene codes for eye color, and this gene may code for the insulin enzyme, and this gene may code for no, not albumin. This gene might code for the skin color. This gene might code for the intelligence of that person. Likewise, there are several genes in the chromosomes, right? Each of these genes separately code for something else. But did you know that the homologous pair has exactly the same gene in the chromosome which is homologous to that? That means these homologous chromosomes share the same gene. If this is for eye color, here also this gene is for eye color. But there is a difference, right? Because that means the genes are the same, but there are different traits of genes. That means there are different varieties of genes. For example, the gene could be the eye color, but you know, there could be black eye color, blue eye color, green eye color, right? So sometimes here the eye color allele can be black, and
Yeah.
here the eye color, here the eye color allele can be brown. That's what you call as alleles. The alternative versions of genes are what you call as alleles. For example, gene is the protein, and for that protein there are different varieties: black eye color protein, brown eye color protein, right? Likewise, there are different proteins. Okay. Now, that is what you call as alleles. That we know. So, if we take a chromosome, if we take this pair of chromosome in our body, there is a pair of chromosome that codes for our blood group, right? That codes for our blood group. Our blood group is actually, our blood group is determined by the antigens on our red blood cell. Let's say the antigen is like a receptor on our red blood cell. Let's assume like that, right? So if we have the A carbohydrate on our red blood cell, it's a carbohydrate. If we have that A carbohydrate on our red blood cell, we say that person's blood group is A. And if we have the B blood group, a B carbohydrate in the red blood cell, we say that person's blood group is B. If we have both A and B carbohydrates in the red blood cell, we say that person's blood group is AB. If we don't have any carbohydrate in our red blood cell, we say that person's blood group is O. So the blood group is determined by these two genes in a specific chromosome, right? There is a specific chromosome which contains two genes like this, right? These two genes are the ones which is coding for our blood group. Let's say the allele over here, the allele over here is IA, and the allele over here is simple I. You know, IA is dominant to simple I. Or that means IA is dominant to simple I. IB is dominant to simple I. Simple I is the according to your syllabus, it's not simple I. According to your syllabus, it's capital I 0. Sorry, IO. Right? According to your syllabus, it's IO. I0 like this, right? You know, IA is dominant. Yeah, IA is dominant to I0. IB is dominant to I0. Not means IO actually. IA is dominant to IO. IB is dominant to IO. IO is the recessive value, right? IO is recessive. IA, IB is dominant to IO. So if we have IA in one chromosome, and if we have I0 in one chromosome, what is the blood group? You know, IA is dominant to IO. Therefore, the blood group is A, right? So such a combination of alleles in the chromosome would result this carbohydrate on the red blood cell, would result the carbohydrate A on the red blood cell. That would give the blood group A, right? Now let's say IB is over here. IB is this allele. Again, you can see IB is dominant to capital I0, right? IB is dominant to I0. Again, then what is the blood group that we are getting? What is the combination of the allele? IB, I0. It is the combination. You know, since IB is dominant to I0, the blood group will be IB. Not IB, the blood group will be B, right? The blood group will be B. That means that those red blood cells will have the carbohydrate B. Now you might be thinking why it's a carbohydrate and not a protein. It's actually a protein, an enzyme which adds this carbohydrate. So when it codes for B, it codes for an enzyme which adds the carbohydrate B onto the red blood cell. That's how it happens, right? When we go into detail, right? So when you have this IB, I0, it codes for an enzyme which adds the carbohydrate B onto the red blood cell so that that person's blood group is B. Now, what if we have IA and IB together? Here we have IA allele and here we have IB allele. We have IA and IB together, right? But actually, we can't be sure, right? That means actually we don't know what is the recessive one or the dominant one. Trust me, both are having the equal power. That means both of them have the equal strength of dominance, right? No one is powerful than the other. Both are having the equal strength of dominance. So what they will do is they will show both their colors together. You could see when IA, I note is there, right? IA overshadowed the I note. It's like when there is a very talented student, right? all the other students, the what do you call, all the other students' colors are not being seen by the teacher, right? Usually the teacher favors the one which is more talented, right? That's how it normally happens, right? But if both are equally talented, if IA and IB both are equally talented alleles, so the teacher would see, okay, both are talented. So so she sees the colors of both the students. Therefore, when IA and IB are together, right, you get AB blood group. You don't see only the A blood group, neither do you see only the B blood group. You see both their blood groups together. So those people have the carbohydrate A in their red blood cell as well as the carbohydrate B on their red blood cell, which means their blood group is AB, right? Their blood group is A. Yes, their blood group is AB. So basically, you can see this is what you called as codominance. Codominance means both are having the equal dominance. But if you took IA, I naught, right? They did not have the equal dominance. IA was more powerful. This is not codominance. Neither is this codominance. This is also not codominance. This is what you call as codominance. Both are having equal dominance. So this is what you call as codominance. And even if you take IA, right, both are of equal powers, right? This is not codominance. This is just homozygous state, okay? This is just homozygous state. So, you know, definitely at homozygous state, the A blood group is shown. And again, IB at homozygous state, B blood group is shown. And again, I naught, you get the O blood group because they are at the homozygous state. This is the only scenario where codominance is shown, right? IA, IB. Right, now let's go for the question and see what they're asking. You can see that they have given, so all what you got to know is IA allele and IB allele are having equal powers. So both of them are shown together. That's it in codominance you got to know. By that you can do a lot of questions. You can see here they have given the blood group and they have given our genotypes. To have blood group A, you know the allele combination or the genotype is, it could be either capital IA or it could be IA I naught, because you know I naught is recessive. So that IA is dominant to I naught, therefore blood group A is shown over here. And if you take B, again IB combination would give the blood group B, or IB I naught combination can also give the blood group B. And AB, there's only one possible genotype. It's IA, IB alleles together. And I and O blood group also, there's only one possible genotype, which is I naught. So they're asking us what is meant by codominance. It's better if you can write the answers given in your that reference book, the textbook that you are using, or else it's better to go with the marking scheme answer. As you can see over here in the marking scheme, they have said in codominance both characters are expressed or both alleles are expressed, right? Both characters are expressed or both alleles are expressed. It's better to say at the heterozygous state. Now, if I was writing the paper, I would write at heterozygous state to explain it clearly. At heterozygous state, because now this is heterozygous, right? You can see A and B are two heterozygous. It's a heterozygous scenario. They are not similar. They are different to each other. So at heterozygous state, when both the alleles are shown, right? When both the characters are being expressed. When both the characters are being expressed, that's what you call as, when both the alleles are being expressed, that's what you call as codominance. When both the alleles are being expressed. If you took IA, I naught, here also it's heterozygous. But are both the alleles being expressed? No. You know, only IA allele is being expressed over here, right? Only A blood group is shown, right? Therefore, this is not codominance. This is what you call as codominance, right? Did you understand that part?
Yes, miss.
Okay, now let's move on to the B part. A man with blood group A. Okay, when they say it's a man with blood group A, it could be IA, or it could be IA, I note. Sorry, IA, I note, right? It could be IA, IA, or it could be IA, I note. And then he marries a woman with blood group B. Okay, he marries a woman with blood group B. Again, it could be IB, IB, or else it could be IB, I note, right? It could be IB, I note. Yeah, it could be IB, I note like this. So they are saying they had 4 children, each with a different blood group. They had 4 children, each with a different blood group. So to do this, you have to take a rough sheet, because that means since you all are still doing your Edexcel IGCSE, you might not have a good practice in doing this, right? Now that's the normal way, right? Now by looking at this, I could say that these two are the genotypes which are getting. That's because of the practice, right? But you have to do it, right? As students, you all have to take another rough sheet and you all have to practice it. You all have to check it out. So let's cross IA, IA with IB, IB, right? Rather than considering these two genotypes, let's first consider these two genotypes and see. Let's wait, I'll take another color. Let's take these two genotypes and see whether we'll get a combination of 4 possible phenotypes, right? Whether we'll get a combination of four different blood groups. So I'll take IA, IA, and I'll cross with IB, IB. But by looking at this itself, you can say, right, you know, the gamete will be IA, right? Here the gamete will be IB. You know, by looking at this, you get only one possible genotype. Only one possible blood group is there if you cross this and this, right? But we want to cross where we are getting 4 different blood groups. Here we are only getting one blood group. So we can say the father is not this and the mother is not this, right? The combination that the father and mother cannot be IA, IA and cannot be IB, IB. So now I'll take a different combination, right? IA, IA crossed with IB, I note. I'll see whether we are getting 4 blood groups, right? Did you understand it? I'm taking this cross now, right?
Yeah.
Yeah, IA, IA, I'm crossing it with IB, I note. IB, I note to see whether we are getting 4 possible blood groups. Actually, you don't have to do like this. You don't have to do it individually. If by looking at it, you can get it if you have the practice. But initially, we require some practice, right? So to show that only, I'm doing it in the basic manner, right? After you get that practice, you can say that the mother is this and the father is this. You can easily understand, right? By practice. But now I'll do it from the basic level, right? So again, in your rough worksheet, try out IA, IA, cross with IB, I note. IA, IA cross. I'm going to cross it with IB, I note. Let's see whether we are going to get 4 different blood groups. Here the gamete is going to be IA. Here what are the 2 gametes we are getting from the mother? IB gamete we are getting. We are getting I note gamete as well. Then let's cross them. IA, IB, and here we are getting IA, I note. Look, we are only getting 2 different blood groups. AB blood group and AB blood group and A blood group. When we cross this and this, so we can understand the IA, IA, right? IA, IA combination is not a favorable combination for the father to give us 4 different blood groups. So I will erase this now because I did it. I crossed IA, IA with both these things, but I did not get 4 different blood groups. So I removed it. Now I know the father's blood group is definitely this, right? Because we found out IA, IA was not there. Now I'm 100% sure that the father's blood group is this. Now we have to find out what exactly is the mother's blood group. To do that, again, I have to do 2 crosses. IA, I note, IA, I note. I have to again cross with IB, IB and C. Okay, I'll do that. IB, IB, I'm going to do that cross. What are the 2 gametes you can get from the father over here? IA gamete and I note gamete. Yeah. What is the gamete you can get from IB, IB? IB gamete only. This is the mother. This is the father, right? Now you are doing the cross. When you do the cross, you get IA, IB combination. And here you get IB, I naught combination, which means here it's going to be AB blood group. Here it's going to be the B blood group. Here it's going to be AB. Here it's going to be the B blood group, which means again we are only getting two different blood groups. But we want to find a cross which will give us four different blood groups, four children with different blood group each. That means four different blood groups. So we know that this is not going to be the mother's blood group. Then definitely the mother's blood group will be this. But let's do it and see, right? We need to confirm it, right? Let's do it and see whether it is exactly that cross that we are looking for. So I'm going to cross IA, I naught with IB, I naught. Okay, so what are the gametes we get from here? I, A gamete, and I naught gamete. From the father, you get I, A gamete and I naught gamete. From the mother, I, B gamete and I naught gamete. Right? Now I'll do the cross. Or you can draw a Punnett square if you like, right? A Punnett square is easy and less confusing, right? Punnett square is drawn like this. You would write the sperm over here, egg over here, and you write the father's alleles over here, IA, I naught, IB, I naught, and you find the combinations. Or else I'll do it like this, since we did everything like this, IA, IB, and then IA, I naught, right? IB, I naught, and I naught, I naught, I naught, I naught. So you can see here the blood group is AB, here the blood group is A, here the blood group is B, here the blood group is O. So you can see this is the cross that gives us four different phenotypes, or basically four different blood groups, right? So what is going to be the father's genotype? That will be IA, I naught. What is going to be the mother's genotype? IB, I naught, right? So you can see by doing the workings in a rough sheet, we have found the genotypes of the parents, right? We have successfully found out what is the genotype of the father and what is the genotype of the mother. So you can see in the marking scheme also, they have given the genotype of the father like this, and they have given the genotype of the mother as IB, I naught. Look at the way they have written the gametes. You know, when you're writing the gametes, they get segregated. The allele combination gets segregated. That means it get divided, right? So you can see, yeah, you can see IA gamete is there, I naught gamete is there. And here you can see in the mother's gametes, here IB gamete is there and I naught gamete is there. Can you see the offsprings that they have, genotypes that they have written? IA, I naught, which give the A blood group. IB, I naught, which give the B blood group. IA, IB, which gives the AB blood group. IO, IO, which gives the O blood group. So you can see by drawing, I think, see, if you draw a Punnett square also, they give you marks, right? You can draw a Punnett square and you can show how you have got these results, right? I'll draw the Punnett square as well. See, I have drawn the Punnett square. Now I'll write the combinations. Look, I get IA, IB. Yes, IB, I naught. IA, I naught. I naught, I naught. I got four different combinations. So I have successfully shown that. So if you also like, you can draw the Punnett square and show, right? And here, transfer error only for gamete mark. That means if you have done a mistake, basically only for the gametes. Transfer the error only for gametes mark. So if you have done an error, but if you have got the gametes correct, I think they are planning to give you mark for the gametes, right? But not for that means sometimes you could have done an error in choosing the wrong parents, right? That means if you have not chosen the correct parents, your gametes would also be wrong, right? But I think they have allowed you to transfer the error only for the gametes. That means here, if you have done an error, when you are getting the gametes, you are doing an error in that also, right? So basically the error is being transferred, right? Did you understand?
Yeah, yes, miss.
Yeah, so since the error is being transferred, but you know the concept, right? You know the concept of getting the alleles, getting the gametes, right? Because of that, they have, if you have done a mistake in finding the parents, they have neglect, that means you won't get any marks for that. But if you have successfully taken the gametes from those parents that you have written, you will be awarded the gamete marks only, right? But you won't be awarded the rest of the marks because the answer would be wrong then, right? So you can say it's quite easy, right? But we need to practice a lot of questions. So this is one type of question that they have asked from codominance. So basically in this genotypes of the parents, you have to write down this is the genotype of the father, this is the genotype of the mother. And in the gametes, you have to show what are the gametes which you receive from the father and the gametes that is received from the mother. And genotypes of the offspring, if you wish, you can draw a Punnett square and show how you have got the genotypes. Or basically, you can write these genotypes separately and show that these four are the genotypes that you are getting. Okay, so that question is done. Now let's move on to our paper today, right? I hope that part is clear. Let's do some more questions separately on a day, taking all the hard questions, right?
Yeah.
Oh, yeah, okay. Now you can see this is a paper 2 question, 2022 June paper I have taken. So you can see here they're asking, give one way that a person can change their diet to a lower risk, of developing high cholesterol. So basically here they have asked us to refer to line 4 and 6. As I told you last week, right, when we were discussing paper 2 paper, I told you you really don't have to read everything over here. But if you like, that means if you are feeling a bit not sure, you can read this part, right? But actually the questions are asked unless they have mentioned the lines. Most of the questions require your knowledge that you have studied during the 2 years, right? Actually they are not they're not targeting, that means if they give a question like this and if they ask you to write down the answer, that would be like an English paper, right? That means doing comprehension, right? So basically here they are expecting you to here they have given you a passage which is related to one of the theories that you have learned, and they're asking from the theories that you have learned. Usually they don't ask questions from the passage, but here they have, if they are referring to the lines, that means you have to go to the passage and then you have to read that part and get the answer. Other than that, the passage is not really important, right? That means by doing questions you will understand, but it's okay, you get only, that means you write your exam only once, so it's okay to read the passage fully if you can manage the time, right? It's okay to read the passage fully before attempting the question. It's okay, it doesn't matter, because you're writing the exam only once. It's okay to have a good idea and go for the questions, right? Now you can see they have asked us, give one way that the person can change their diet to lower the give one way that the person can change their diet to lower their risk of developing high cholesterol. So they have asked from line 4 to 6. You can see this is line 4 and this is line 6. You can see they have mentioned the treatments include lifestyle and dietary changes to help you remain as healthy as possible. Medicine is also used to control associated problems such as high blood pressure and cholesterol levels. Other treatments need to be used for severe kidney diseases when kidneys stop working. These are dialysis and kidney transplants. So they have asked us one way the person can change their diet to lower the risk of developing high cholesterol. So it's not something that you should take from the passage. It's something that you should tell from your knowledge. How can you reduce the risk of having high cholesterol? You can intake less lipids or fats, right? You can take in you can take in less lipids or fats or oils. Either one of them is correct. Or else you can have a balanced diet, right? If you are having a balanced diet, you might not get high cholesterol. Or else you can say take less foods that contain cholesterol. Take less foods that contain cholesterol. For example, you can write down an example as well, because then you will further explain what are the foods that contain cholesterol, right? You can mention eat fewer eggs that contain cholesterol. Eat fewer eggs that contain cholesterol. So you can see it's not an answer given in this passage, but they are referring to that, they are asking you to refer to the knowledge of what you have had during these, what you have learned during these 2 years to answer this particular question. And if you have just written eat less cholesterol or eat fewer eggs, they are not giving marks for that, right? That means if you have directly said something like eat less cholesterol, it's not correct. You have to take less food that contain cholesterol, right? You have to mention about food. Take less food that contain cholesterol, or take less lipids, or take less fats, or take less oils, or have a balanced diet. So be a bit careful when writing the answers. That means I will check your answers. Did not have time to check the answer, right? I will check your answer and tell you where you've gone wrong or anything like that. You can see the way that you have to write the answer is you have to mention about the food. If you did not mention about food, right, that means it's okay. Here they have not given marks for food. But if you just write take less cholesterol, that won't be awarded any marks. So it's always better to write a full answer for any question that you've been given, right? Okay.
So written as the person can consume a diet with less fat in it.
Yeah, that is correct. A person can consume a diet with less fat. That is a good answer. Less fat, less oil, less lipids. It's good. But if you just say eat less cholesterol, like eat, use the word eat and write less cholesterol, that's not good, right? Okay. So you can see one function of the kidney is excretion. Another function is osmoregulation. Yeah, so in your syllabus also, the kidney has two functions. One is excretion, the other is osmoregulation. What do you mean by osmoregulation? Osmoregulation means maintaining a constant environment. Maintaining a constant environment when it comes to water and solutes in your body. For example, you can see if you take our body cells, right? If you take our body cells, if they have a high water potential than the solution outside, the solution outside is blood plasma, right? The solution outside is blood plasma in our cells. So if our blood plasma have lot of solutes, like you are eating lot of fast food, right? And you have lot of salts in your body now, right? You have intake lot of salts, so your plasma will be concentrated with salts, right? Therefore you can see your plasma doesn't have lot of free water molecules. The water potential outside is low. The solute potential outside is very high. Such a solution is what you call as a hypertonic solution. So you can see water potential inside the red blood cells is high, therefore water will move out. Water will move out and the red blood cells will shrink, right? Here they have said shrivel, or the red blood cells will shrink, right? And when they shrink, their function is inhibited, right? So that's going to be dangerous for us. That is why we have osmoregulation. It will help us to maintain a constant solute concentration outside, right? Such that the water potential outside is equal to the water potential inside. So by osmoregulation, they are trying to keep an isotonic solution. Osmoregulation is a part of homeostasis. What do you mean by homeostasis? Homeostasis means maintaining a constant internal environment in our body. So osmoregulation is an example of homeostasis, right? So you can see it always try to maintain our body cells in an isotonic solution. And sometimes we might drink a lot of water, right? Sometimes many people drink a lot of water, right? More than 4 liters, sometimes 6 to 8 liters, they drink water. So in this case, what will happen to them? The water potential outside will be high. When the water potential is high outside than the water potential inside, water will move in and the red blood cell can burst, and this will be fatal, right? This will be fatal. Yes, and that is why we hear some news like the person has drunk a lot of water and that person has died. That's because osmoregulation has not functioned properly in their body, right? Okay, you can see now, they have asked for the definition of osmoregulation. You can write maintains water level, right? Or maintains water of the body. Maintains, the word maintain should be there. Maintain, or else you can write control as well. Control is also given mark. Maintains or control the water level of the body. The water level of the body or water level of the plasma, right? Or else you can write down like this also. Maintains the salt and water levels. Maintains the salt and water levels in the body fluids. Maintains the salt and water levels in the body fluid. Salt and water levels in the body fluid. You can write down it's a process which maintains the salt and water levels or the salt and concentrations in the body fluids, right? Or in the cells, in the body fluids or in the cells. If you have written down salt and water, and if you have the word maintain or control, you get 2 marks for that answer. Right. Now let's move on to the next question. Here they have asked us, explain why a person with severe kidney disease will need dialysis to continue for life. So, you know that dialysis is a machine that we use, right? Give me a second. Give me a second. I will come soon. Give me a second. Okay, so you can say dialysis is an artificial machine which acts like a kidney. When your kidney is damaged, there are 3 things, with one kidney you can survive. But if both of your kidneys are if both of your kidneys are damaged, right? You either, that means if you either will have to die if you didn't do any treatment, or you can do a kidney dialysis, or else you can do a kidney transplant. Either 3 of these, right? These are the 3 options we have for if both our kidneys are damaged. So dialysis means, right, your unfiltered blood is taken before it goes to your kidney. They are taking that blood from your renal artery, right? From an artery, they are taking that blood, right? From an artery, they are taking that blood before it goes into the kidney, and they are passing that blood through a dialysis machine, right? I think I got an image of the dialysis machine. Yeah, see, I think, see, you can see this is the dialysis machine. You are taking the blood out like this. You can see you take the blood out like this, and there is a pump. You pump the blood into this dialysis machine, and you can see in the dialysis machine a dialysis fluid is allowed to go into this dialysis machine. In this dialysis fluid, it has normal glucose concentration. That's because when it has normal glucose concentration, just like in the blood, here this is the blood solution which is coming from inside. When you have normal glucose concentration, there won't be net diffusion of glucose molecules, right? Because the reason why we are doing dialysis is not to remove any glucose molecules, right? We want to remove the excretory substances like urea, the bad substances, right? Ammonia, right? We want to remove the bad substances from our body. That's why we are doing dialysis. We don't want glucose to be excreted from our body, right? Even in our normal kidney urination, that means in our normal urination also, we don't release glucose unless we have diabetes, right? So here we should maintain the dialysis fluid should have normal glucose concentration, which is similar to blood. And also it should not have any urea, right? When it doesn't have any urea, here the blood inside, I'm coloring it in brown. The blood inside has high levels of urea, right? The blood that we are pumping, right? It has high levels of urea. So high concentration of urea is inside the dialysis, inside the blood. And you know, inside the dialysis fluid, there is zero urea, no urea at all. Therefore, low concentration of urea is there. So you know, from a high concentration to a low concentration, urea will diffuse into the dialysis fluid. Urea will diffuse from the blood into the dialysis fluid. So urea in our blood will be removed by dialysis, right? So that is the function of dialysis. You can see you send the fresh dialysis fluid, and after the urea gets collected in that dialysis fluid, it is being removed back, and you can see the blood which is being contaminated. Any questions?
No, miss.
Oh, okay. The blood which is being contaminated enters in, and because of diffusion, urea passes into the dialysis fluid, and again purified blood gets removed from the dialysis fluid. That's how our blood gets purified using the dialysis fluid. So here they are asking, explain why a person with severe kidney disease will need dialysis to continue for life. They're asking us to go through line 15 to 16. When they have asked us to go through 15 to 16, we must obey them. So let's go and see what they have mentioned over there. If the patient does not have a kidney transplant, treatment with dialysis will usually need to continue for life. So that is line 15 and 16. They're asking us to explain. When they say explain, usually they require justification. But in this case, you don't have any data to justify, right? Because if they have given us a sample of data, then you can take that data and justify. But here, since they have not given us any data, you don't have to do any justification. You just have to that means you just have to explain it to them. Explain it, sort of like give a description about it, right? How you are going to do that means why a person will need to do a dialysis, right? You are just going to explain. That is the meaning of the word explain. But there are some scenarios where the word explain means it asks you to take the data which is being given in the tables or in the graphs, right? So in such cases, explain means you have to take that data. But here explaining means since you don't have any data to explain, you have to use your knowledge and explain. So basically, you can say, right? You can say the body produces toxins, right? The first point should be the body produces toxins. So you are going to explain it to the examiner. Why we need a dialysis? That is because the body produces toxins. Or else instead of toxins, you can write down the body produces urea, or the body produces, oh yeah, the body produces metabolic waste, right? Either one of these three you can write. The body produces toxins, the body produces urea, the body produces metabolic waste. The next point is, now when both our kidneys are damaged, right, there is no way of excreting them, right? There is no way of excreting them. So you can write down these needs to be excreted, right? Because there's no way of excreting them now when our kidneys are damaged, right? So these somehow needs to be excreted. You have to write that meaning. These needs to be excreted. These needs to be excreted, or else instead of that, you can write down they will become toxic if they remain in the body, right? Instead of, for the second point, you can mention why we need to, that means you can mention what is needed to be done with this metabolic waste. What is needed to be done? You need to excrete them, or you can mention that they will become toxic if they remain in the body. They will become toxic if they remain in the body. If they remain in the body. As the third point, you can write down the kidneys cannot. That means here they are asking for only 2 points. So if you have written any 3 of these points, you can get the mark. As the third point, you can write down the kidneys cannot recover, right? That is why you are going for dialysis, because the kidneys cannot be recovered when both the kidneys are being damaged. It's not curable unless you go for a transplant, right? So the kidneys are, the kidneys cannot recover, or the kidneys are uncurable until you do a transplant. So basically, they are asking why a person with severe kidney disease need to do dialysis. First point is now see, you are producing justification, right? you are giving some justification. That is because the body produces metabolic waste and that needs to be excreted. And you know, the kidneys won't recover on its own. When both the kidneys are damaged, they won't recover on its own. Therefore, you need dialysis. So you provide some justification and then you write, therefore, we need dialysis. So explaining means giving some justification for the question that they have asked, right? Okay. Now, let's move on to the D1. The peritoneum acts as a partially permeable membrane. Explain what is meant by a partially permeable membrane. Lines 23 to 24. Let's read lines 23 to 24. 20, 21, 22, 23, 24. Inside the abdominal cavity is a natural membrane lining called the peritoneum. This membrane is partially permeable. So the answer is not given in the paragraph. You have to mention what this partially permeable means. Partially permeable means it only allows some molecules to pass through it. Only allows some molecules or substances to pass through it. Right? Only allows some molecules or substances to pass through it. Or else, and also that is the first point that they have given, which is equivalent to 1 mark. And the next point you must write is a partially permeable membrane does not allow or does not let large ones or large molecules to pass through. Does not allow large ones or charged ones. Right? The plasma membrane also does not allow large ones or the charged ones to pass through. Right? To pass through. So these are the 2 points that you have to write. 1 point is it only lets or only allows some molecules or substances to pass through. The other point is it does not allow large ones or charged ones to pass through it. Right. Then we can see the next question they're asking. Explain why the dialysis solution must contain purified water, glucose, and mineral ions. You know, when the dialysis solution contains water, right, the water concentration in the dialysis solution is very high. So when you pass blood, right, when you have blood over here, and you know now just let's assume the water potential of the blood is higher than the dialysis water potential, then blood will move out of our body. The water will move out of our body. Water will move out of our blood. That's not good, right? We need to retain our water. In excretion, we are only excreting excess amount of water that we are having. We are trying our best to retain the water because water is essential for us, right? So we are only excreting the excess amount of water that is in our body. So it's essential that the dialysis solution, right, should contain purified water. Not just that, it should also have glucose. Because when glucose is of similar concentration to the blood, you know, there is no net movement of glucose in or out from the blood. So glucose concentration is also maintained. And mineral ions also, by excretion, you are not planning to excrete any mineral ions because mineral ions are required by our body. In case if we have excess mineral ions only, we are excreting them, right? And that's also due to a disease or something. So normally we don't excrete any mineral ions from our body, right? Therefore, you can say in order to not allow minerals to be minerals to be removed from the blood only, we maintain a good concentration of mineral ions in the dialysis fluid. So how are you going to put it into words? They have asked us to refer to line 19 to 21, not refer to. That part is given in line 19 to 21. So let's read that and see as well. You can see line 19 to 21. The composition of the dialysis solution is water, glucose, and mineral ions at the same concentration that occurs naturally in the blood. Can you see the water concentration, the glucose concentration, and the mineral ions concentration are equal to as that of in the blood. That is because they don't like any net diffusion of those substances into the dialysis fluid from the blood, right? Otherwise, if we lose them, that's going to be a disadvantage for us. So we are not going to allow them to go anywhere, right? So you can see, you can write it down like this, right? So that, right, the reason, now you're going to explain, right? What is the reason why we are maintaining same concentration of these substances in the dialysis fluid as well as in the blood? So that those substances will not leave the blood, right? So that those substances will not leave the blood. Those substances will not leave the blood. Will not leave the blood. Will not leave the blood. And leave the blood by what? Leave the blood by diffusion. That is another point, right? By diffusion. Now assume, right? Assume in the dialysis fluid we have very low concentrations of water, and in the blood we have high concentration of water. So blood will move out. Blood will move out from the, sorry, not blood, water will move out from the blood into the dialysis fluid by diffusion, right? So the word diffusion, they have given a mark for the word diffusion. Here one mark, here one mark, right? And then you can further write it down. The reason why what is the reason why you do not want those substances to leave out? That is because our cells require those substances, right? Our cells, you can write down the importance of these substances. Our cells require water, right? Require water for metabolic reactions, right? That is why we don't like to excrete them. Our cells require water for metabolic reactions. And you can write another point. Our cells require glucose for respiration, right? Our cells require glucose for respiration. So why are we going to excrete them? There's no point of excreting them, right? That is why we maintain the glucose concentration in the dialysis fluid similar to that of our blood, right? And also, if you know a function of mineral ions also, you can write down our cells require mineral ions for this and this purposes, right? For example, we need iron to produce more hemoglobin, right? If you know an example of mineral ions, you can write it down. We need calcium ions, right? Our cells require calcium ions to maintain strong bones and teeth, right? You can write down something like that. But I think better than, yeah, I think better than that point, it's better to write the water, the function of water and function of glucose, right? Because they have allowed you to give a correct function of mineral ions, but it's safer to write down it's easy, not safe, it's easy to write down a function of glucose and function of water, right? So if you have written any two of the 4 points, you can get 2 marks, right? Now you can see here they have asked us, explain how the composition of the dialysis solution results in the waste products being removed from the blood. If I draw the dialysis machine like this, outside you have the dialysis fluid, inside you have the blood. So in the dialysis fluid, there is no urea at all, or else you can write down low concentrations of urea are present in the dialysis fluid. But inside the blood, there's going to be high concentrations of urea, right? Because the blood has traveled from different parts of your body and collected the urea, excreted from your cells, and finally it comes into the dialysis machine to get purified, right? So here, there's going to be high concentration of urea. So we should maintain low concentration of urea in the dialysis fluid. Then only we build a concentration gradient, right? So that from the high concentration region of urea to the low concentration region of urea will pass out of the blood into the dialysis fluid. Urea will pass out of the blood into the dialysis fluid. So the first point that you have to write is have low concentration of urea or low concentration of waste products. All right, you can write down low concentration of urea low concentration of urea or low concentration of waste products. Low concentration of waste products in the dialysis fluid. In the dialysis fluid. In the dialysis fluid, right? So that the urea, right? So that the urea will move out by diffusion, will move out into the dialysis fluid. You have to mention the word diffusion, right? Will move out into the dialysis fluid by diffusion, into the dialysis fluid by diffusion, by diffusion, right? Low, this is the first point. The low concentration of urea or low concentration of waste products are there in the dialysis fluid, so that urea will move out of the dialysis fluid by diffusion, right? The word diffusion is very important over here. You can also mention by a high concentration to a low concentration by diffusion, right? You can further explain. See, can you look at, can you see the way that you have to answer the explain questions? You have to provide a lot of details. You have to give the best shot. That means you have to include all the things that you know. Here also you could see when they ask about the why the dialysis solution needs to contain purified water, why it needs to contain same amount of water in both the blood and the dialysis fluid. We even went to describe the function of glucose and water, right? We went to describe the function of glucose and water in the cells. That's because when they say explain, they require a lot of justifications from us, right? So we have to write everything that is, if you write all the accurate information in your knowledge, you will easily be able to get the marks that is allocated for the question. But the information needs to be accurate, something that you have learned, right? So it's better to write a lot of that means all the theories in your head which are accurate, right? And all acute, that means for explain questions, there is nothing like to the point. That means answers are not always to the point, right? Answers are that means there are so many answers. Answers are starting from one point and you could carry it out to some other point as well. For example, look at this question. You can see in this question here they have asked why dialysis solution must contain purified water, glucose and mineral ions. And they have asked us to go through line 19 to 21. That's because here they have mentioned that they are having same amount of, see, the composition of dialysis solution in water, glucose and mineral ions is at the same concentration that occurs naturally in the blood. From there we understand in the dialysis fluid and in the blood, we have same concentrations of water, glucose, and mineral ions. So we have to explain the reason why it's like that. So you can start from one area. You can say, so that those substances will not leave the blood. That is the major point that hits your head, right? So that water, glucose, mineral ions won't leave the blood by diffusion. You say, you further explain, right? Now imagine you forgot to write by diffusion, you might have not got that mark, right? If you forget to write diffusion. So when they say explain, it's better to explain it into detail itself, right? Leave the blood by diffusion, right? So you include a detailed point by diffusion from a low high. It's okay to even write if your handwriting is okay, that your handwriting is okay. I saw your handwriting. It's okay. You can write a lot of things using that handwriting from a high concentration to a low concentration or down a concentration gradient. It's okay to write that point also, right? That means to be
I couldn't answer those two questions.
You didn't hear me.
No,
You didn't answer.
I couldn't answer those two questions.
Yeah, you did. Yeah, I did not check your what do you call paper. I checked your handwriting. That means I went through the paper like from, that means from the surface, and I just saw your handwriting, right? So your handwriting is sufficient to write all those things. That means to fit all the information into these spaces. That's what I meant, right? Okay. So by diffusion, down a concentration gradient, right? Down a concentration gradient. And also further you can write down the importance why you should not allow the glucose molecules to leave the blood. What is the importance? Because glucose is required, right, for cell respiration. If it moves out, then it's going to be a big disadvantage for us. And water is important for what? Water is important for metabolic reactions. So you can say when they say explain, you have got so many things to write because it requires justification. So these words are very important. Now let's move on to the next question. A person is using APD. It's a word given in the diagram, given in the passage. You can say calculate the percentage of their time used for treatment in a week. So what is this APD? What are the lines? Line 33 to 35. Okay, so you can see 33 to 35, another form of PD, automated peritoneal dialysis requires a machine to fill and drain your abdomen. 3 to 5 exchanges are performed during the night while you sleep. The whole process lasts for a total of 9 hours each night. So, they are saying this APD process each night, right, it takes 9 hours. Each night, it takes 9 hours, right? The APD process each night, it takes 9 hours. They're asking us to calculate the percentage of the before that, I want to ask you the reason why you didn't do this question. Is it because dialysis was like you all have not done dialysis, or was dialysis a bit difficult?
It was a bit difficult. I couldn't understand like what the was happening.
Okay, so did you understand now? Do you have any doubts?
No, miss, I understood now.
If you have any doubts, you can always ask me, right? Let me know. Okay. So you can see they have done the APD each night. They have done it for they're doing it for 9 hours each night. So they are asking us to calculate the percentage of their time which is used for treatment in a week. The percentage of the time used for treatment in a week. So 9 hours means per day. One day, that means one day they are doing it for 9 hours, right? Each night means one day, right? That means in a day they are doing it for 9 hours, right? So 9 hours per day. How many days are there for a week? There are 7 days for a week. Yes, there are 7 days for a week. And how many hours are there? That means they are asking us to calculate the percentage. So how many hours are there in a day? 24 hours are there in a day. So how many hours are there in a week? 24 hours multiplied by 7, right? This is the total number of hours per week. 24 hours multiplied by 7. This is the number of hours you do the AP dialysis for a day, right? AP dialysis, you do it for 9 hours per day. And to calculate the time total time for 7 days only, I multiplied it by 7. So you here also for one day you have 24 hours. To calculate the total time for the week, you multiply it by 7. And you multiply it by 100 to calculate the percentage. So you can see 7 and 7 get cancelled off. And it's 9 over 24, right? You get 37.5 percentage as the answer when you simplify. 9 over 24 will become 0.375. Multiply by 100, you will get 37.5% as the answer. You can write down the answer like this, or you can round it off as well and give the answer as a whole number to 38%. Either one of these, you get you get 2 marks, but you need to show the calculation. To show that you are dividing 9 by 24, you will be awarded 1 mark. For your final answer, you will be awarded 1 mark. That is why in this syllabus, it's really important for you to mention your workings. I studied the AQA syllabus also. So I remember in the AQA syllabus, they're not requiring these, workings. But in your syllabus, they're requiring the workings, then they're giving marks for your workings as right, and also for your final answer, right? Now you can see, suggest why people may prefer to use CAPD instead of hemodialysis, right? Let's go through 31 to 32, right? 31 to 32. I think it's better if in this case it's better to read this whole paragraph, because you have a lot of extra information and you won't be afraid if you read the whole paragraph. But in some certain situations, it's okay. Go with reading the paragraph. Always when you get the paper, read the paragraph, or else first read the question. Now what I've been practiced was, I first read the question and I go for the paragraph. Then it's easy for me. I read all the questions, so the questions are in my head, and when I'm reading, I understand the connection between the question and the paragraph, right? So you can do it in any method you like. Anyhow, all what you got to do is apply your knowledge to each and every question. So here they asked us to go through unit lines 31 to 32, right? So 31 to 32, a typical one form of PD, continuous ambulatory peritoneal dialysis, does not require a machine. So they are saying a form of peritoneal dialysis called CAPD, right? A form of peritoneal dialysis. Again, it's a dialysis done to the peritoneum, okay? Done near the peritoneum. That's what you call as peritoneal dialysis, right? So there is a special form of peritoneal dialysis called as CAPD, continuous ambulatory peritoneal dialysis. Now they say it does not require a machine and it is possible to work around with the dialysis solution in your abdomen. I think you might have seen that there are some people in movies and all, right? That means if I draw the human like this, they are carrying some bag or something in their stomach, right? They're carrying, yeah, that is the dialysis machine, right? Otherwise you have to attach that, otherwise that person needs to be in the hospital throughout his lifetime until the kidney transplant is done, right? But there are small children who needs to go to school and everything. I remember, yeah, there are small children who needs to go to school, so they use the CAPD method where they can carry the dialysis machine, and when they come home they have to remove that. Again they have to put a good new solution to continue the dialysis, right? So basically, having CAPD instead of the normal dialysis method, instead of homeodialysis method, what is the advantage? They have asked, suggest why people prefer CAPD instead of the normal dialysis. What is the reason? You can walk around. Otherwise, you've got to be in that hospital room for around, like, until you do the kidney transplant, right? And it's expensive as well, and you can't walk around with that. You have very big machines attached to you, so that's really troublesome, right? Because of that, people prefer CAPD, because the answer is there in line 31 to 32. That is, the reason is because you can walk around, right? You can walk around, or else you can write down. It can be done at home. You don't need to be at the hospital. You can walk around, or it can be done at home. Suggest means, the word suggest means you can write your own thoughts. You have to be a bit creative and think out of the box too. It's okay to write down what is in the in the 2 lines. See, they have given that mark if for writing you can walk around, but also you can be a bit creative and you can write down a reasonable answer. Something like you can stay at home and do it, right? The CAPD method. So you can stay at home. You can stay at home and it can be done at home. It can be done at home. Can be done at home, right? Or else it does not require machinery. It does not require machinery like in dialysis. You just attach a bag into your abdominal region and you just carry it out, right? It does not require machinery. Yes. I have seen a movie where the girl gets bullied because she has that bag, and when she runs and all that, what do you call that? That bag makes different sounds, and once someone did something to that bag, and that bag exploded or something like that, right? I remember watching a Korean movie like that. So CAPD is done in real life. You can see, so that is the answer. You can write. You can walk around. It can be done at home, or else it does not require machinery. 3 of those, either one of those 3 answers, you get 1 mark for writing. Then the G1, they have asked us, describe how the substances in a human kidney result in the correct substances being retained in the blood. So You know, in our human kidney, there is a structure called nephron, right? We have a structure called the nephron. The nephron has a structure like this. This is the Bowman's capsule, right? These are the proximal convoluted tubules and the distal convoluted tubules. And this is the loop of Henle, right? You know, here there is a big ball of capillaries called as the glomerulus. They're asking us how the structures in the human kidney result in the correct substances being retained in the blood. You know, here in glomerulus, substances pass into the Bowman's capsule by a method called as ultrafiltration, right? This method is called ultrafiltration. There is a neuron, there is an arteriole called the efferent arteriole. Here there is an arteriole called the efferent arteriole. So this brings blood, this brings blood into the glomerulus, and this carry the efferent arteriole carries blood away from the glomerulus. And I will tell you something, this efferent arteriole is thinner than the afferent one. Afferent is very thick, efferent is very thin. That's because when lot of blood passes through it, only a small amount of blood will get away, right? That means lot of blood will pass into the glomerulus, but slowly only the blood will get away from the glomerulus. That means we are keeping the blood inside the glomerulus for a long time because of this adaptation. But that's not really required for you to know. Here I'm just explaining it in detail. You can see the blood is kept for a long time in the glomerulus so that a process called ultrafiltration will take place. In the ultrafiltration, you know, large protein molecules, right? Large molecules, large protein molecules, red blood cells, white blood cells, they are not going to pass into this into the Bowman's capsule because they are very big molecules. What's not going to pass into the Bowman's capsule? RBCs, red blood cells, white blood
Protein.
cells, proteins and platelets, right? They are not going to move into the Bowman's capsule. Only the soluble substances like glucose, right? Even glucose will pass inside, okay? Glucose and mineral ions, right? Urea, right? The water, right? These are the substances which will travel into the Bowman's capsule. But wait, if glucose pass into the Bowman's capsule, it's going to be excreted. We don't want glucose to be excreted like that, right? We need glucose, right? That means we need glucose for aerobic respiration and everything. But see, it's going into the Bowman's capsule. That means it's going to be excreted soon. We should prevent this somehow. How are we going to prevent it? Do you know this efferent arteriole over here forms a network of capillaries like this called as peritubular capillaries, right? These are called as peritubular capillaries. No need to know the word, called peritubular capillaries, right? So basically, you can see glucose will again move back into the capillaries by a method called selective reabsorption, right? The glucose concentration in the capillaries is very low, but the glucose concentration in the blood, in the Bowman's capsule is very high. So down a concentration gradient, it's going to be selectively reabsorbed back into the capillaries. So you can see glucose will get selectively reabsorbed back into the capillaries, right? But urea, just because urea concentration inside is high, it won't get selectively reabsorbed, right? That is a separate mechanism. They don't allow urea to be reabsorbed back into the capillaries. Only glucose molecules are allowed to go back into the capillaries, right? And you can see this happens in the proximal convoluted tubule over here, right? In the PCT, right? And also in this collecting duct, okay, there is a structure called the collecting duct over here. In the collecting duct, you know, see, I told you water comes in, right? Water gets into the Bowman's capsule by the glomerulus. So will water also be excreted by our body? If water is going to get excreted like that, see, all our water is now inside the Bowman's capsule. If all our water is going to get excreted like that, there is no point of drinking water. So when we drink water, all that water will be excreted, right? Only some amount of water should be excreted. So most of the water should again be reabsorbed. So in the collecting duct, water will be reabsorbed back into the capillaries, right? So can you see how the structure of this is the interior structure? You know, in the kidney there are nephrons. This is one nephron that I have taken. There are millions of nephrons in the kidney. So you can see how the nephron structure, right? How the nephron structure has been adapted in such a way that means to allow the correct substances to be retained in the blood. Otherwise, if glucose leaves through excretion, that's going to be dangerous, right? We won't have glucose for aerobic respiration, right? But remember, this is in a healthy person. In an unhealthy person, definitely glucose will be excreted if that person is going to have diabetes. In a healthy person, glucose will be retained in the blood. Glucose will be taken back into the blood. Right. So how do you write the answer? You have to start from the glomerulus. You have to describe, right? When you describe, you have to describe the whole process of this filtration which is taking place inside the kidney. So they are saying describe. When they say describe, you have to give an account, right? I sent you a small table where the words are there. In that they say to give an account, right? When they say describe, you have to give an account, and the statements in the response needs to be developed. That means you have to tell it like a story, and there's no need of justify. You don't have to say why is glucose getting reabsorbed. You don't have to justify. You just have to describe what is happening over there. That's it. So tell it like a story, right? Okay. So how what is the first thing you can say? Proteins, right? Proteins, or else you can say large molecules, they are not leaving the glomerulus, right? Or else you can, in brackets, if you want to write, you can write down red blood cells, white blood cells, platelets, proteins, right? You can write all these things, right? So they are not going to, they won't leave the glomerulus, right? They won't leave the glomerulus. So you can see they are retained in the blood, right? Because if they leave the glomerulus and if they enter the Bowman's capsule, they will be excreted, right? So because they are large, they won't enter they won't leave the glomerulus. Or you can write down in the converse manner, they won't enter the Bowman's capsule, right? Something like that. That is the first description. And then you can write down, glucose enter, right? You can say glucose enter. You won't get any point for writing that. You can write glucose enters the Bowman's capsule, right? Glucose enters the Bowman's capsule. But here you don't get any point. After I write the description, I'll tell you what are the points, what are the sentences which gets the point. But I'm doing the description first. Glucose, glucose enters the Bowman's capsule, but it will be reabsorbed. It will be reabsorbed back into the blood. It will be reabsorbed back into the blood. Back into the blood. Back into the blood. Where? I'm writing the location. By the proximal convoluted tubule. Or in the proximal convoluted tubule. In the proximal convoluted tubule. In the proximal convoluted tubule. And further I'm writing water is also reabsorbed. Water is also reabsorbed in the collecting duct. So all the useful substances are not excreted, they are retained in the blood. We have answered the question. What is also reabsorbed where? The location in the from the collecting duct. From the collecting duct. So these are the sentences which gets the points. You can see proteins, large molecules won't leave the glomerulus. You get 1 point for writing that. Glucose enters the Bowman capsule, no points for that. But if you write it will be reabsorbed back into the blood, you get 1 point. And if you write the location where it is being reabsorbed, you get another point. And if you have mentioned what is also getting reabsorbed by the collecting duct must be there, you get 1 point. So if you have written any of the any three out of these 4 points, you will be awarded 3 marks. That means just to have a flow only, I mentioned that glucose enters the woman's capsule, right? So you can also add things to give it a flow, right? But there are certain things which will be awarded marks because they're asking us how the structures in the kidney result in the correct substances being retained in the blood. So we have mentioned the structures and we have also mentioned the substances which are being retained. Proteins, glucose, what are the substances which are retained in the blood and are not excreted from our body. Some water will be excreted, but most of the water is retained. Right, let's move on to the second question. Here they have done an experiment. A student uses this method to measure the energy value of a sample of bread. So he measures the mass of the sample of the bread. He put 20 cubic centimeter of water into a boiling tube. Then he placed the thermometer. He record the initial temperature. Then he placed the bread sample of bread on a mounted needle like this. And then he light the bread using a Bunsen flame, and he quickly moved the burning bread under the boiling tube. And if the bread stops burning, you again relight the bread using the Bunsen flame, and you again place it back under the boiling tube, and you repeat this until the bread does not burn, basically until it turns into ashes, right? Then only it won't burn, right? So you record the final temperature of the water in the boiling tube. So so they have done an experiment like this. Let's see what are the questions that they're asking. Here you can see the table shows some of the students' result. Okay, so here they have given us the initial temperature, final temperature, and the change in temperature. What is the change in temperature over here? That will be 15 degrees Celsius. Final, final subtract the initial. And energy released in joules and energy released by 1 grams. Let's find it out when we are doing the questions. So you can see in the A1, they're asking us to calculate the energy released from the burning bread. The student, to calculate the energy released from the burning bread, the student uses the formula energy is equal to mass of water, specific latent heat of water, which is 4.2 grams, and increase in temperature. Then they calculate the energy in joules released by 1 gram of bread. So this is the formula which you use to calculate the energy released by energy released by that sample. Okay, energy released by the sample of bread that they have taken, right? This is the sample of bread that they have taken in the third experiment, right? 0.20 grams. Then using that later, we are going to find out what is the energy released by 1 gram of bread. First of all, we have to find what is the energy released by 0.2 grams. Then using the ratio method or using some method, we can find out what is the energy released by 1 gram, right? So let's do you can see they are initially asking us to calculate the increase in temperature for the third sample of the bread. So even though they have given us 1 mark, it's better to show that you are subtracting 36 from 21 and getting the answer, right? I think it's safe, right? That means Edexcel IGCSE papers always requires workings. Even though they have not given marks this time, it's better to show them how you have done it, right? It won't take a lot of time, right? Just writing 36 minus 21, right? You can say calculate the energy released in joules by the third sample of bread. So they have given us the formula, right, to calculate the energy released. So basically, what is the mass of water in grams? Okay, mass of water in grams. So basically, now here they have, even though they have said mass of water, we are considering yes, 0.2 grams. 4.2 multiplied by 50. Yeah. Now, mass of water, you can see here they have put it in grams. But the bread sample here they have given, okay, so basically the mass of the bread sample is 0.20 grams, right? Actually, we are but here can you see they are asking for the mass of water in this equation, right? But we don't know what is the mass of water in this equation. In actually, that means we have to use the density method, right? Density is equal to mass over volume. Density is equal to mass over volume. The density of water is 1 gram per cubic centimeters. The volume of water, have they mentioned what is the volume of water? Yes, 20 cubic centimeters of water, right? 20 cubic centimeters of water. So this is how you find the mass of water, right? So you have to use the density. See, can you see they have asked for the mass of water over here, right? Now this is very important in physics also, right? When they give us mass of water, we have to find what is the mass of water. For that, we should know what is the density of water, right? Density of water, you need to know this, but here they have given it. See, 1 cubic centimeter of water has a mass of 1 gram, right? What is the density of water? 1 gram per cubic centimeter, right? 1 gram per cubic centimeter is the density of water. So, you know, density is equal to mass over volume. So, what is the volume of water that we are using for the experiment? 20 cubic centimeters of water, right? So, 20 cubic centimeters, you have to multiply by 1 gram per cubic centimeter. So, what do you get? You get 20 grams of water. Otherwise, you don't have to use this density equation. They have said 1 cubic centimeter of water has 1 grams, right? So how many cubic centimeters of water are you using? You're using 20 grams.
20.
Yes, you're using 20 grams, right? So basically that is the mass of water that you're using. Energy is equal to 20 grams multiplied by, usually if they have not written this in grams, you have to write it in SI units. Okay. If they have not mentioned in grams, you have to write it in kilograms. But since they have mentioned it's in grams, then you can write it down in grams. Otherwise, we have to use SI units to calculate the energy released in joules. But maybe here they have given 4.2. It cancels off with this unit. 4.2, that specific latent heat cancels off with the grams. Okay. Okay, no need to worry about that too much. I'm just saying that we will get different types of questions in the future, right? So you have to be a bit careful with the units each time. So 20 grams multiplied by 4.2, you have to multiply it by 15 degrees Celsius. The answer will be 1260 joules. Okay, so you know the 1260 joules was released for 0.20 grams of bread, right? It was released for 0.2 grams of bread. Now we have to calculate the energy released by 1 grams. Can you see it's the ratio method? For 1 gram, for 0.2 grams, if it is 1260 joules, then for 1 gram, how much is that? Basically, you have to multiply this by 5, right? 0.2 multiplied by 5, you get 1 grams. And here you can multiply it by 5, you get 6,300 joules, which is released for 1 grams, right? Energy released by 1 grams will be 6,300. Right, now that is done. Then they have said the energy value given on the packaging of the bread is 10,400 joules. But look at the value that we got. We got 6,300 joules. But the bread, 1 gram of bread actually has 10,400 joules. That means we have done some mistakes in our experiments. That's why we got a lower value than the actual value, right? We have a very big deviation. That's because we might have done some experiments. There can be some unreliable things that we have done in this experiment. So they are asking us to comment on why the students' method give a different value. When they say comment, they require us data to form a judgment. They usually requires the data. You can see, you can first mention 6,300 joules is lower than 10,400 joules. Or else you can say students' value was much lower. So you are referring to the data. Look, when they say comment, initially you refer to the data and give the description, right? You refer to the data and then only you form the judgment. So first you refer to the data. What is the data? The student's value is lower than is much lower than the actual value, right? That is the first point, right? That is the reference to the data is much lower than the actual value, right? Now you give the judgment, right? What is the reason? You suggest things now, right? When they say comment, you can suggest the reasons. First you give reference to the data and you say this is what has happened. Students value is lower than the actual value. Then you give the comments. That means you give what could be the reasons that this happened, right? So one reason might be because all the energy might not have been released from the food, right? Just because you could say we are burning the food, right? When we are burning the food to release the energy in the food. So sometimes the food might not have been burned completely. That might be the reason why it has not given sufficient energy, right? So it could be, we are suggesting things, right? So it could be correct, it could be wrong, doesn't matter. These are the possible answers, right? So all the energy, not all the energy is released from the food. We could say not all the energy is released from the food. Is released from the bread. Is released from the bread. In other words, you can write down it is not fully combusted, right? The bread is not fully combusted, to release all the energy. That's why we got a lower value. If it was fully combusted, we would have got the original value, right? Or at least a value which is closer to 10,400. But our value is completely different. Maybe it's because it's not fully combusted. And also, let's say it has been combusted fully, but we could say the energy is not all the energy is not transferred to the tube. You know, when you are burning like this, right? Not all the energy which is produced from this bread will go directly into the tube, right? Lot of energy will radiate, right? Lot of energy will radiate to the outside environment, to the surrounding. So what we are calculating will definitely not be the total energy in the bread. The answer will be wrong, right? Definitely, right? The answer will have a lot of deviation. Why? Because a lot of energy is radiated when we do like this. There's no insulation or anything like that, right? So because of that, there's a lot of energy radiated. Yeah, so you can say energy or heat is not, all the energy or heat is not transferred to the tube. Is not transferred to the tube. Is not transferred to the tube, right? Or else you can say that the energy is lost. Energy is lost into the atmosphere. Energy is lost to the atmosphere. To the atmosphere. They have not mentioned surroundings. I don't know why. They have mentioned atmosphere, right? Energy is lost to the atmosphere. And also you can see here when you are burning a glucose molecule, right? Energy is emitted as light as well, right? Not only as heat, right? That means the energy stored in the glucose molecule is not completely given as heat, right? That's light energy as well, right? So some of the energy might have been given out as light, because when the Bunsen flame is lit up, in order to light up the Bunsen flame, in order to give light energy, some of the energy is in the glucose molecule is given up as light. Therefore, energy is lost as light also, right? Energy lost as light. Energy lost as light. And also, right? And also the reason could be, right? Sometimes the person might not have stirred this the water in the boiling tube. So when the water is not stirred, it might not give the accurate temperature, right? In order to give a uniform constant temperature, the water should be stirred. So you can write down water might not be evenly heated, or water might not have been stirred. That could be an answer, right? Water might have not been stirred. Have not been stirred. Not just that, another possible thing, you can write any possibilities. They might have had a big gap between the Bunsen flame, right? Between the flame and the tube, right? So you can see there's going to be a big distance. So not all the heat is going to touch the boiling tube. So the distance might not have been fixed. right? The distance might not have been a fixed one, or the distance might be too big, right? So due to these reasons, you can write any of those reasons. First, you have to mention the data. That means when they say comment, you have to, they require the data from it. What is the data? The data is student's value is lower than the actual value. Then you make the judgment, right? Why is the student's value low? You give some suggestions, right? So if you have given at least 4 suggestions along with this particular statement, you can get 5 marks, right? Okay, now let's move on to the next question. They're asking, give 2 ways that the student could modify to apparatus, modify this apparatus to achieve an answer nearer to the energy value given on the packaging of the bread. Now, you can see here a lot of energy is being radiated to the outside. So why can't we insulate this area? That means why can't we, that means why can't we use a heat shield or something so that energy is not radiated to the outside, right? We can use a heat shield, right? So that energy doesn't escape to the outside environments, right? So you can mention use a heat shield, right? Using a heat shield, use a heat shield. Use a heat shield. And also here, to maintain constant temperature inside the water, you can use a stirrer, right? You can use a stirrer, right? To maintain an accurate temperature inside the water, use a stirrer, right? Use a stirrer. And also you can insulate this tube, right? If you can use a if you can insulate this tube, the heat which is radiated from this tube can also be reduced, right? Because when this tube get very hot, right, not all the heat is used to boil the water, right? Not all the heat is used to increase the temperature of the water, right? Some of the heat gets radiated from the tube surface. So you can say use a insulated tube. Use a insulated tube. Or else you can use a lid, right? Use a lid to prevent heat from, that means sometimes the water can get evaporated, right? By absorbing a lot of heat energy. If water gets evaporated, the mass of the water that we have calculated will be wrong, right? Here the mass of water we calculated as 20 grams. If water gets evaporated, right, then the mass of water that we are calculating is wrong, then our final answer will definitely be wrong, right? So it's better to use a lid to prevent evaporation, right? So you can say using a lid, right? Using a lid. And also you can keep to keep equal distance. Now sometimes your hand might be shaking, right? So you will have different distances, right? From the tube to the mounted needle, you will have different distances throughout the experiment. So why don't you use a clamp and a stand? Using the clamp and the stand, you can hold the test tube in a fixed position, and you can hold the mounting needle at a fixed position, right? So the distances won't change, right? Throughout the experiment, you are keeping a fixed distance, right? So you can say fix the position of the foot or the clamping needle, or use a stirrer or stirring needle, or use a stirrer or stir the contents, or else you can write use a heat shield, use a lid, or an insulated tube. These are the possible answers for this question. Right, now let's move on to the next question. The diagram shows a cell found in the lining of the human intestine. You can see these infoldings that you see in our small intestine is our villi, right? So if you magnify a villi, this is how the villi actually looks like, right? This is how the villi looks like. These cells over here in the villi are called as the epithelial cells, right? They are called as these single cells. You can see they are 1 cell thick, right? 1 cell thick, right? Can you see only 1 cell layer is there? Those cells are epithelial cells, right? And they have magnified the epithelial cells. On the epithelial cells, they have again a lot of infoldings called as microvilli, right? Called as microvilli. So basically here they have taken an epithelial cell and here they have shown us the microvilli, right? Epithelial cells are the cells which are found in this villus, right? Villus is the singular word for villi. Right. So they are asking us which of the following label structures is the microvillus. Of course, the C is the microvillus, right? D is the plasma membrane. A is the nucleus. B is the mitochondria. Therefore, C is the answer. Which of the label structures produces ATP? You know, mitochondria is the structure which produces ATP, which is the powerhouse of the cell, which carries out aerobic respiration. Therefore, answer will be B, which is the mitochondria. Right. Then you can see these cells form the lining of the small intestine, as you can see over here. Right? They are the ones which form the lining of the small intestine. They asked us explain how the structure of the small intestine is adapted for absorption. So basically, you can see, explain how the structure of the small intestine is adapted for absorption. The small intestine is folded, right? The small intestine is folded like this. Why is that? That is to increase the surface area. Yes, to increase the surface area. It's folded to increase the surface area. To increase surface area. Then you can say it contains many villi also, right? It contains many villi. You can see it contains many villi over here. So many villi. Why does it contains many villi? Again, to increase the surface area. No issue. You can write it down. Contains many villi to increase the surface area. To increase the surface area. And also, in the villi, you can see in the villi they have epithelial cells. In the epithelial cells, they have microvilli. Again, what does that do? That also increases the surface area. So you have to explain the structures, right? Contains microvilli, right? Contains microvilli, right? Contains microvilli again to increase the surface area. To increase the surface area. Yeah, surface area. And do you know, if you take one villus like this, right, inside the villus, there are blood capillaries. Okay, there are blood capillaries. Not just that, there is a structure called the lacteal, right? Into this lacteal only, now you know, give me a second, right? I have been called moment, I'll take one. Okay. Now, if you take the lacteal, you know, in the small intestine, triglycerides or else lipids, right? I'll take it as lipids. Lipids are being broken down into their smallest units, right? Smaller substances. You know, the smaller substances are glycerol and fatty acids, right? Glycerol, fatty acids are the smaller substances in the small intestine. In the what do you call? In the ileum, right? Not in the ileum, jejunum and ileum, jejunum and, give me a second, ileum, jejunum, duodenum, yeah, duodenum, right? In the duodenum, they have been broken down into fatty acids and glycerol, right? And in the ileum and the jejunum only, they have been absorbed, right? They have been absorbed, right? I suddenly lost my memory. So basically, you can see fatty lipids have been broken down into fatty acids and glycerol. So, do you know that these lipids, they cannot be absorbed by the blood capillaries? There is a special type of tube, right, which is a part of the lymphatic system inside the villus called as the lacteal, right? They are the ones which absorb these fatty acids and glycerol molecules into them. Let's say other substances like amino acids, what else? Glucose molecules, right? Vitamins, these are being absorbed into our blood, right? These are being absorbed into our blood capillaries, but fatty acids and glycerol are being absorbed into our lacteals. So you can write down, they contain lacteals to absorb lipid, right? They're asking what are the structures in the small intestine, right? which is adapted for absorption. So lacteals are there. You can say lacteals are there inside the villus. If you want to go into detail, you can write down lacteals are there in the villus in order to absorb the lipids, right? Lacteals are there in the villus, or lacteals to absorb lipids. Lacteals to absorb lipids. And they have capillaries. They have blood capillaries to absorb glucose and amino acid. To absorb glucose and amino acid. And what else? Can you see? This is one cell thick, I told you, right? The epithelial cells are one cell thick. What is the reason why they are one cell thick? To allow for fast
diffusion distance.
Yeah, to shorten the diffusion distance, right? Yeah, they have given that answer, to shorten the diffusion distance, so that there is a short diffusion distance. You can write down there are thin walls or one cell thick, right? Or the walls are one cell thick. One cell thick for fast diffusion or short diffusion distance. For short diffusion distance, right? For short diffusion distance. Now, so if you have written those points, you will be given 4 marks. Then you can see cells in the human placenta also have microvilli. So, if you take the human placenta, right, you know, human placenta is the one, human placenta looks like this. In the human placenta, there is the umbilical capillaries like this. Umbilical capillaries are there, and they are formed from the umbilical arteries and the umbilical veins. So basically, here the blood of the fetus is there. Here, that means here the blood of the, not the fetus, the blood of the mother is there. Here the blood of the fetus is there in the umbilical capillaries like this, right? Here you can see like an interstitial fluid. The blood of the mother is there surrounding the villi, surrounding the microvilli. The blood of the mother is there. And inside the microvilli, right? Inside the microvilli, there are blood capillaries of the fetus. The umbilical capillaries, you call them, right? Umbilical capillaries of the fetus are there. So basically, what is the role of the human placenta? The role of the human placenta is, now you know the umbilical, you know the umbilical artery, what does it carry? The umbilical artery carries the waste products, not like in humans. In humans, so you know the artery is the one which carries the good stuff. But in the fetus, umbilical artery carries the bad stuff, right? That means the waste products. So let's say this is the, I'll draw a clear image. Yeah, so basically umbilical artery carries bad stuff and umbilical vein carries the good stuff, right? Yeah. So if I draw the placenta like this, right? Placenta like this. This is the mother's blood. Okay, mother's capillaries are there like this. Mother's blood vessels are like this. And you know, from the capillaries, mother's blood leaks out, right? Mother's blood leaks out. And this is mother's blood, right? And these are the veins, right? These are the veins of the mother's blood. This is the veins. The red one is the artery, right? And here, if you take in the placenta, the brown color structure, right? They have microvilli. Not just that, they also have the umbilical capillaries like this, right? They have a network of umbilical capillaries, okay? A big network is there. And do you know there are structures called, there are 2 umbilical arteries. These umbilical arteries, now here is the fetus, okay? Somewhere here it's the fetus, F-O-E-T-U-S, right? Fetus is there. Attached to the fetus, only the placenta is there. From the fetus, I'll say this is the fetus. I don't have space to draw the fetus. So basically, I'll say this is the fetus. Okay, from the fetus, 2 umbilical arteries are coming. Right, 2 umbilical arteries are coming from the fetus. These umbilical arteries, right? Not 2 umbilical arteries are coming. 2 umbilical arteries are attached. And the umbilical vein is attached like this. Umbilical vein is attached. From the umbilical vein, right? That means here, here in the mother's blood, there is oxygen, right? So oxygen will diffuse into the capillaries like this. And through the umbilical vein, oxygen will get into the fetus. Can you see? Usually veins carry deoxygenated blood in humans, right? But in fetus, the umbilical vein carries oxygenated blood. Umbilical vein, okay? There is only one vein which carries oxygenated blood in our human body. That is the umbilical vein. And that's also in fetuses, right? In fetuses, in the tiny babies which are in the mother's womb, right? So basically, here they carry the oxygenated blood. And right, here the fetus, right? The fetus, they transport the waste products like carbon dioxide, urea, right? They transport it through the umbilical arteries. Usually, what does arteries do? Arteries usually carry oxygenated blood, right? But in this case, can you see the arteries, the umbilical arteries are carrying the waste products, right? You don't have to know about umbilical arteries and veins in detail when you're writing the answer to this, right? But I'm just trying to explain the process, right? So you can see carbon dioxide and urea, they will diffuse out, right, from these capillaries. And the good blood will come from the mother. That means not good blood. Oxygen will come and then oxygen will move through the umbilical vein into the fetus. So that is the importance of the placenta. The reason why we have a placenta like this is to also to make sure that the mother's blood is not getting mixed with the fetus blood. If they mix, the fetus will die, right? If they mix, there will be a reaction. There will be a coagulation process. That's not in your book at all. You don't have to know anything about that, right? So if the babies and the mother's blood mix, there will be a process called coagulation, and that will lead to the death of the fetus. Sometimes it's fatal for the mother as well, right? So their blood does not mix. You can see the placenta separates their blood, right? So I will erase this, and this is the explanation. So they are asking the role of the human placenta. One role is the oxygen can be transported from the mother to the fetus. That is one function, right? So write down all the functions. When they say describe, write down everything you know, right? Oxygen from the mother to the fetus. Oxygen travels from the mother to the fetus, from the mother to the fetus through the placenta, to the fetus through the placenta. Not just that, digested food, nutrients, right? Nutrients, digested food, or nutrients, they also travel to the fetus from the mother, travel to the fetus from the mother through the placenta, from the mother through the placenta, right? And also, the placenta is needed to remove the waste products, right? So to remove the waste products, you can write down that also. Remove the waste products. See, now when I was explaining you, there was a very big content, right? About what you call the umbilical arteries, veins, right? But they are not referring to that. They're just asking us for the role of the human placenta. So you just have to mention what is being done. It removes the waste products. If you want, you can be specific and write down urea, carbon dioxide from the fetus to the mother. From the fetus to the mother. And what else? The antibodies, the hormones, right? Antibodies, they can be transported from the mother to the fetus. Antibodies are needed for good immunity, right, in the baby. So antibodies from the mother's blood is transported into the fetus using the placenta. Not just that, in reproduction, when we are learning about the hormones, we know that the placenta can also produce progesterone, right? So you can write down that is another role of the placenta. Placenta produces the hormone progesterone. Right. Now, any doubts?
No, miss.
All right. Now, yes, I will get into this cloning part, since that means since we don't have enough time, because this cloning part is highly important, right? That does not mean I will not discuss the other parts. When we have a SEP today, I'm going to my boarding house also. That is the reason I don't have enough time to do it. After your class, I'll be moving into my boarding house today. My vacation is over. So because of that, I'll move into the 6th question directly, because it's a good question, the cloning part, right? Okay, so you can say scientists can now produce farm animals by cloning. Since Dolly the sheep was born in 1996, many different species have been cloned. Okay, so Dolly was the first animal to be formed by cloning. Now they are saying the form of cloning is used is called somatic cell cloning. The first ever clone of a champion racehorse was announced in 2005 in Italy. The foal was cloned from Pierce, a world champion in long distance horse races. Okay, so they are asking in the first question, describe the stages scientists use to clone a male horse. So there are two types of cloning processes. One is the adult cell cloning. Okay, one is called as I'm talking about the theory, adult cell cloning, and the other is called as the embryonic cell clone. Right, in your syllabus you have two types of cell cloning. What are those? Adult cell cloning and embryonic cell cloning. Right, when they say they are using a male horse, they are using an adult over here. They are not using a little baby, right? They are using a horse, which is a male horse, means that is an adult. Okay, so here they are using the adult cell cloning process, not the embryonic. So from these two processes, by looking at the question, I understood what is the cloning process that they are using. Right, they are using the adult cell cloning process. So in our syllabus, we are learning what is the adult cell cloning process. Right, have you done that in school?
Yes, miss.
Okay, so basically the adult cell cloning, in the adult cell cloning process, right, if you take in the adult cell cloning process, but this is what basically you are doing, right? You are taking, and now you are, that means let's say there is a horse like this. There is a horse, right? From a horse, you are taking a sperm cell, right? You are taking a sperm cell from, that means you are taking an egg cell, not a sperm cell. You are taking an egg cell from the horse, right? You are taking an egg cell from a horse and you are removing the nucleus of that egg cell, right? So the egg cell has a nucleus, right? You are removing that nucleus from the egg cell. You remove it, right? Now the cell doesn't have a nucleus. Now the cell has all the other things like the mitochondria and all those things. The egg cell will process, but it does not have a nucleus, right? And you take this male horse, right? You take this male horse. You take that, now here the egg cell could be taken from anyone, right? Egg cell could be taken from anyone. The reason of taking an egg cell is to allow it to be divided, because these gametes can be easily divided rather than normal cells, right? That is why you take an egg cell. So you take egg cell from anyone that you like, not from anyone, from a horse you take it, and from that male horse, from this male horse, you are taking a skin cell, right? You are taking a skin cell, any body cell that you like, right? Basically, it could be a skin cell, or else you can write down a body cell. You are taking a body cell. So from this horse, you took a body cell. Now that body cell has a nucleus. You're not going to remove that nucleus, okay? That body cell has a nucleus, right? And this egg cell, you remove the nucleus, right? You remove the nucleus. So here, what you are trying to do is, you are inserting this body, you are inserting this body cell, right? You are inserting this what do you call it? Inserting this DNA in this body cell into this egg cell, right? You are taking this DNA out of the body cell and you're trying to insert into this egg cell, right? And you're doing that using an electric shock. You can do that, right? Giving an electric shock, you can do that. So basically, not by an electric shock. There is a method called DNA recombination method, right? Using that method, DNA technology, it's a DNA technology. You can basically insert this, right? Not recombination. Using DNA technology, you can insert this particular DNA in the body cell into the egg cell. Now, this DNA is inside this egg cell now, okay? This DNA is inside the egg cell now. Now, what do? You apply an electrical shock, right? You apply an electric shock. Then this egg cell will start to divide by mitosis, right? The egg cell will start to divide by mitosis. Now, this egg cell doesn't have its own nucleus, okay? The egg cell has the nucleus of that body cell that we have taken, right? Because its own nucleus was removed. So it has the nucleus of the other cell now. Now it's going to divide with that other nucleus, right? So many identical cells will be formed, right? So many identical cells similar to this particular egg cell, similar to this cell will be formed, right? And then you take one of those cells, right? You take one of those cells, right? Actually, similar to the means it, wait, like this, right? When it divides mitotically, it forms a ball of cells, ball of identical cells like this, right? Ball of identical cells like this, something like this is formed. So you take this ball of identical cells and you take a surrogate mother, right? You take a surrogate mother host, right? And you can put that egg, right? You can put that egg, that not the egg, you can put this ball of cells into that mother's womb. So this mother is a surrogate mother. She's not the real mother, right? Yes, here you are doing asexual reproduction. You're not using the sexual reproduction method. Cloning is a method of asexual production. It is a asexual reproduction method, right? So here, this is called the surrogate mother because it's not the biological mother, right? Basically, you don't have a mother in this case, right? So you are using the surrogate mother and you are implanting that ball of cells. So in that mother's womb, the baby will develop and later this mother will give birth to the baby. So that's how a clone is formed, right? So you can see that's how an adult clone is formed, right? Adult clone is formed in that way. So you can see this, that means the adult, the clone, the baby which is formed, it is identical to this male host. The baby which is going to be formed, right, when it grows big, right, when it ages, it will look exactly like this male host, exactly identical to the male host, because you take the nucleus from that male host and you insert into that egg cell and only you allow it to divide, right? So all the DNA in that male host is also in the baby's DNA. So when the baby grow big, right, he will exactly look like the male host, right? So they're asking us to describe the stages scientists use to clone a male host. So basically they are referring to the knowledge that you have to describe it. What is the first step? The first step is you are taking the nucleus, right? You are taking the nucleus from the cell of a male host, from this male host, right? You are taking the nucleus from the body cell of a male host, right? You are taking that nucleus and you are inserting that nucleus into an egg cell, right? Because that egg cell's nucleus should be removed first, okay? First you can start like this. I don't like to go with the marking scheme because it doesn't have any pattern actually. So it's better to go like this, okay? I'll tell you how to go through it. Then it has a pattern, flow. Then it makes sense. First you can say, take an egg cell, right? Take an egg cell or take a enucleated egg cell. Take an enucleated egg cell. Enucleated, that means the nucleus is removed, right? Take a enucleated egg cell. Okay, that is the first one, right? Then the second point is take the nucleus. Take the nucleus from the body cell of the male host. From the body cell of that male host, see, of this male host, body cell of this male host. And then you can write down, insert this nucleus into the egg cell, right? That is the next point. Insert the nucleus into the egg cell. Then you have to mention you're using an electric shock, right? Use the electric shock or use electricity, right? Why are you using electricity? To stimulate the cell division by mitosis, right? So you can write it down like this. The marking scheme they have given us, they don't give us the whole description, they give us the important points, right? So when you're writing, you can write it down like this. Electric shock is given. If you want, you can write it down. I read. Electric shock is given to stimulate the egg cell to divide by mitosis. Electric shock is given to stimulate the egg cell. An electric shock is given to stimulate the egg cell to divide by mitosis. An electric shock is given to stimulate the egg cell to divide by mitosis to form an embryo. to form an embryo. For writing embryo, you get another point. To form an embryo. Then you can write this embryo is inserted into the womb. That is another point. This embryo is inserted into the womb. Womb. This embryo is inserted into the womb. Right? This embryo is inserted into the womb. Right? Of a surrogate mother. Of a surrogate mother. The word surrogate should be there. Then only you will be awarded the other mark. To a surrogate mother. The embryo is inserted into the womb of a surrogate mother. Then Pyrrhus, I think it's the name, was castrated, had his testicles removed at a young age. Right. Explain why this stopped him reproducing normally, but did not stop him being used to produce a foal by cloning. So you know, when the testicles are removed, he can't carry out sexual reproduction, right? So when they say explain, you have to, that means they are requiring justification in your answer, right? So why can't he reproduce normally? Because no sperms are produced when the testicles are removed, right? No sperms are removed. When testicles are removed, there's no sperms produced. I mean, sperms, yeah, when testicles are removed, no sperms are produced. So therefore, there's no fertilization. That means he can't impregnate a female, right? He can't impregnate a female. And then you can write down, why he could produce a foal by cloning. Now you gave the reason why he can't reproduce normally. Then you have to give the reason why he could reproduce by cloning. Because cloning, you know, that means in cloning you use the DNA from the body cells, not from the testicles, right? You use the nucleus from the body cells. You are not damaging any body cells of the fetus, right? You are basically removing the testicles only, right? So removing the testicles won't affect the cloning process, right? So he can still carry out cloning, because in cloning you could see we are taking the DNA from the body cells, right? Not from the testicles. So he could carry out cloning in the normal manner. So you have to write down, explain why this stopped him reproducing normally. The reason is no sperms are produced when the testicles are removed. In the testicles only, the sperms are being produced, right? So you can write down, when the testicles are removed, no sperms or no gametes are being produced. No sperms or no gametes are being produced. No sperms, no gametes means there won't be fertilization. No fertilization. Or in other words, you can write down, he can't impregnate a female. He can't impregnate, right? He can't get a female to be pregnant, right? He can't impregnate a female, right? So we have given the reasons why this has stopped him from reproducing normally. Now we have to give the reason, the justification why he still can reproduce by cloning. That is because in cloning you are using the body cells, right? In cloning you are using the normal body cells. In cloning, the nucleus comes from the body cells. So cloning is not affected by the removal of the testicles. In cloning, the nucleus comes from the body cells, right? The nucleus that we are using comes from the, not the testicles, comes from the body cells, In cloning, the nucleus comes from the body cells. The nucleus that we are using in cloning is coming from the body cells. So that is the answer. So you got 3 points. You have to write all these 3 points to be awarded the 3 marks, right? And you can see the embryo cloning, right? Now we discussed about the adult cloning. I'll give you a summary of the embryo cloning as well. So you can see in the embryo cloning, right? In the embryo cloning, I think, yeah, in the embryo cloning, basically, you do artificial fertilization, right? You take a cow, which is very good. Let's say you want to produce a clone which can produce a lot of milk, right? So you want to produce a cow which produces a lot of milk. So you are going to take the best cow, right, which produces a lot of milk, and you are going to take the best bull. that bull also produces really tasty milk, right? So you're getting the best cow and the best bull, right? And then you're taking the cow's eggs and you are taking the bull's sperms and you're fertilizing that, artificial fertilization or in vitro fertilization, right? And this will form an embryo, right? This will form an embryo. You know, the embryo cell will start to get divided. That means the embryo cell starts getting divided, right? When the embryo, that means when the embryo cell starts getting divided, first after the zygote is formed, zygote only develops into the embryo. Later, this embryo starts getting specializing, right? That means an eye is formed from one cell, a leg is formed from one cell, a hand is formed from one cell, a heart is formed from one cell, right? So before it starts to get specialized, we are taking these embryonic cells. Actually, these are embryonic stem cells, right? We are taking these unspecialized cells, and you can see we are inserting them into the host mothers. We are taking some mother cows, right? And we are inserting part of that embryo into each of the parents, simply in vitro fertilization, right? What we are doing for humans also. So it's also but it's not cloning, right? Because in that you are not taking that mothers, you are not taking the babies, the embryo, and you're not going to divide into parts, and you're not going to give it to several mothers, right? You're only going to give it that to that specific mother, right? But in animals, you're going to take that embryo and you're going to give it to several animals, several mothers. So basically, they are not the real parents also in that case, right? That's why it's called as cloning, because at the end, a lot of identical individuals are formed. But in human in vitro fertilization, it's not cloning, because you're only producing one individual, one baby, right? You're not producing many babies identical to each other, right? Okay. So here you can see that you are putting it in. So here you can see the embryo, the zygotes, you have divided them, you have splitted the embryos like this, and you're putting it into the new parents, right? You're putting it into the new mothers, and those mothers will give rise to calves which are genetically identical to each other. That's called as embryo cloning, and this is what you refer to as adult cloning. So that is the difference. Now, here they have asked, suggest why horse racing does not allow the use of non-natural methods of breeding, including cloning. Why doesn't horse racing allow? Now, you have to suggest. Now, they are not giving these in your textbook and all, right? That means the suggest questions, most of the suggest questions are not in your textbook, right? You have to think and write. Why this horse racing doesn't allow cloning? That is because, now think, you are it's one of you, right? Now you and your friends are doing horse race, right? You are the owner of a very fast horse, right? That horse is really fast, but someone's going to clone your horse, right? When they clone your horse, they get an identical horse just like yours, right? It will take some time for the horse to grow from a baby to an adult, but somehow when it grows into an adult, it will be as powerful as your horse, and by that time your horse might be old also, but that person's horse is young, and he has the same genes just like your horse. So when that friend of yours who has cloned your horse come to the horse racing competition, would you like her to win the competition? No, that's cheating, right? Because she has cloned your horse, right? So it's not ethical, right? That is why the horse racing, right? Horse racing doesn't allow cloning, because they are artificial animals, right? They're not produced by the natural means. They're artificial, and you can do a lot of cheating using that, right? The real champion will not be awarded at the end, right? Basically, you're stealing the genes from an organism and you're producing another organism identical to that. So that's not ethical, right? One reason is it's not ethical, right? You can write down. One reason it's not ethical, right? It's not ethical, or it's you can mention if you have given the idea of unfairness or the idea of unequal competition or the idea of cheating, you'll be awarded that mark. Or else if you have and also the embryonic cloning method, is it good to take an embryo from a mother and divide it into several embryos and put it into different mothers? Now, would a human mother like, when they do in vitro fertilization, would the human mother like her embryo being taken, being divided into like 4 to 6 segments and giving that embryo pieces to different mothers? Our parents would never like that, right? That means taking their embryo and giving it to different parents, right? So there are ethical issues with the use of embryo. With the use of embryo also there are ethical issues, right? Because it belongs to one parent. How can you do like that, right? Without any consent, right? So there are ethical issues with the use of embryo. An embryo means a life. So you are dealing with a life. So ethical uses of embryo is there. And also, basically, uh, when you have cloned animals, right, they are genetically identical to each other. So basically there's no variation in that horse race, right? You're doing a race, you're having a competition to see who is the best. If all are equal to each other, if all are having equal talents, right, then there's no point of doing the horse racing, right? There's no variation, right? So you can write down, to maintain genetic variation only, you're not including cloned animals in horse racing, right? In horse racing, to maintain genetic variation. So if you have any idea like that, you'll be awarded this 1 mark. Then they are saying, state one difference between cloning an organism and genetically modifying an organism. So cloning an organism will basically, right, cloning an organism will basically give the same genetically identical offsprings. Everyone will be genetically identical to each other. But when you are genetically modifying, you are giving a variation to that particular organism. For example, you are taking a tomato plant and you are inserting a gene which will allow that tomato to basically stay ripe for that means stay ripe for around like 2 to 3 weeks, right? So you are varying the genes, right? You are modifying the genes, right? So when you're modifying the genes, there is a genetic variation. But cloning, it gives rise to genetically identical offsprings, right? So you can write down, clones are identical to each other, right? Or they have the same genotype. Clones are identical to each other, or they have same genotype. You can continue writing, genetic modification produce genetic variation. Genetic modification produces genetic variation, right? So these few questions I'll be discussing during the next day. The ones we have, that means last week also we had that red blood cell part. So I'll take a separate day to discuss these questions along with the hard areas that you got, right? But anyways, I badly wanted to teach the stem cells part today because there's a lot of theory involved in that part. So I will see. Yeah, so I will be calling it a day today. If you have now you can see the that means your exam year is starting, right? That means within 2 days your exam year will be starting. So it's better on the new year, right? Now we don't have any time for celebration. I know it's good to celebrate, but after another year you can definitely celebrate, right? The new year very happily, right? So basically during this new year, my advice is, right, this is what I used to do during my O levels and A levels. So basically during the new year you can eat whatever you like, you can enjoy a bit, but it's better to be with the books these days, right, rather than deviating, right? So on the not on the Christmas day, on the New Year's Day, right, at 0000, that means at 12 o'clock usually we are awake, right? So you can stay awake while doing some questions, right, while doing some either biology questions or any questions, physics questions, maths questions. Usually I used to do maths a lot at this time every year. Still, even to this year I'm going to do some work at this time, right? I don't have much work, but I'll be doing something because it's like a positive thing, right? You're at the start of the new year, you're doing something related to your studies, so basically you'll be involved with studies, right? So try to that means this week try to make it productive. You can do past papers and you can send it to me. so that we can discuss those past papers, and also you can collect some questions after you've done the past papers, and you can send those to me so that we can discuss those areas, right? So I will send you a timetable of what I'm planning to do. Then you have an idea about the direction that we are going, right? So for the month of January, I will send you what we are going to do during this month. So if you have any amendments, you can ask me to change those, right? Okay. So
Okay.
I will be finishing it for now, and study well, right? Okay.
Thank you.